All right, welcome to the second lecture of chapter 23, and we're still going to continue on looking at how the kidneys work. We're going to start looking a little more specific at some of the functions of the parts of the kidney today. But as always, let's go ahead with our not really attendance questions. First, what are the components of a nephron in order of the path of filtrate? Next, what is a renal corpuscle? And finally, what aspect of blood pressure do kidneys directly influence? So go ahead and pause the video, try to answer those before listening to the answers. Now let's see what the answers to these should be. What are the components of a nephron in order of the path of filtrate? So we produce filtrate at the glomerulus. We're going to see how that happens. Once that filtrate is produced, it moves into Bowman's capsule. And then we move into the proximal convoluted tubule, the PCT, into the descending limb of the loop of Henle, then the ascending limb of the loop of Henle, into the distal convoluted tubule, the DCT. And some sources, that's all that they include in the nephron, but really the collecting duct, the CD, is how I will classify the nephron. The collecting duct is what we will consider the final segment of the nephron. And now the collecting duct, actually several different nephrons connect to it. So each nephron will have Bowman's capsule, PCT, the full loop of Henle, the DCT, but then many of those together will connect to one single collecting duct. Next, what is a renal corpuscle? We mentioned this in the previous lecture. Renal corpuscle is both the glomerulus, that modified capillary, plus Bowman's capsule. And what aspect of blood pressure do kidneys directly influence? Kidneys directly influence blood volume. So blood volume. Now, remember back when we were talking about blood pressure, I said don't flush that information from your head, because when we get to kidneys, really kidneys are blood pressure part 2. Sure, kidneys cleanse your blood to make urine, but kidneys directly influence blood volume, which influences blood pressure, and that's going to be a huge topic several times through the next few lectures. All right, so let's get back to our PowerPoint. Last class we were talking about first just general formation of urine, and we began talking about the anatomy of the renal corpuscle. Here in this image, we can see the afferent arteriole that brings blood in. We can see the glomerulus here inside where that filtration is taking place. The blood that is not filtered leaves through the efferent arteriole. But as we pass through the glomerulus, let's think about what the glomerulus actually is like. It's rather like a garden hose that somebody has taken a nail or a pin or something and poked a lot of holes in. So imagine if that was the case and you turned that garden hose on and let water flow through it. What would ultimately happen? That water would come spraying out of the garden hose. Well, remember the glomerulus is a fenestrated capillary. It's got holes in it. So as that blood passes through the glomerulus, liquid sprays out of the blood, and there's some stuff dissolved in that liquid. It's very similar to plasma, but with less protein and a few different ions than plasma has. But that filtrate, which is what it now is, collects here inside Bowman's capsule. And as Bowman's capsule fills up, that liquid, that filtrate, passes into the PCT and on through the rest of the glomerulus. So today, everything that we look at is going to be concerned with how does the glomerulus actually filter the blood. So here is an image from your textbook in page 898. but we're actually going to draw this through. So let's go ahead and get a piece of paper and get ready to draw this. And I've already got the image here, so I'll give you a moment to kind of draw this out and get caught up. But while you're doing that, I want to talk about how I asked everyone to go back and review capillary fluid exchange. I told you that today's lecture would be very, very similar to that lecture on capillary fluid exchange. So what I've drawn here, this red tube is a capillary, and over on the left is the afferent arteriole bringing blood in. Over on the right is the efferent arteriole carrying blood away, and the capillary in between, in this case, is the glomerulus. And remember, glomerulus is inside of Bowman's capsule, so that's what I've drawn here in blue. Now, think about capillaries. Normally, the layout of a capillary is an arteriole brings blood in and a venule drains blood out. That's not the case here for the glomerulus. Another modification in addition to those fenestrations is that it is flanked on each side by an arteriole. That allows for constriction because remember, arterioles are very muscular. That allows for constriction and dilation on each side to kind of maintain those pressures that are common in a capillary. So let's look to see what these pressures are. And they're the same pressures, we're just giving them slightly different names than we used before. We've got blood inside the glomerulus, inside this capillary, and blood will press against the surface of its container, trying to press its way out, and we call that hydrostatic pressure. Now we're going to call that the hydrostatic pressure of the glomerulus. And here, this is found to be 60 mmHg. We've also got things like albumin and other solutes in the blood. Remember, solutes suck, they pull blood or pull liquid towards themselves. In the case of blood, it's pulling that liquid towards itself. And it's got a pressure of 32 mmHg in the opposite direction. So I'm calling that negative 32. Hopefully this is ringing a bell back in capillary fluid exchange. Well, now we are pushing liquid out into Bowman's capsule. So Bowman's capsule is filling up with liquid, and that liquid is pressing against the walls of its containers, pressing against Bowman's capsule, but it's also pressing against the capillary from the other side. It's pressing its way into the glomerulus. So it's got a pressure of 18, and again in the opposite direction, so we'll call it negative 18 mmHg. But that leaves one more pressure, osmotic pressure of Bowman's capsule. But there is no osmotic pressure of Bowman's capsule. It's 0, and in fact, your book doesn't even list it. So why would that be? Why would we not bother listing the osmotic pressure of Bowman's capsule? Well, Bowman's capsule doesn't have any proteins in it when things are functioning correctly. Remember, proteins are much too big to be filtered, so there should not be any proteins out here in Bowman's capsule in order to provide that osmotic pressure. So we don't even list osmotic pressure of Bowman's capsule. Now, if we add all of this up, everything together, we get our net filtration pressure, or NFP. And net filtration pressure of Bowman's capsule is 10 mmHg. We are filtering liquid out of the blood under a pressure of 10 millimeters of mercury. But remember when we did capillary fluid exchange, there was pressure down here on the arterial end and another pressure down on the venous end. But there's not a venous end here. These arterioles work in concert to keep the pressure the same throughout the entire glomerulus. Remember, in the capillaries that we looked at before, we filtered on one end and reabsorbed in the other. Well, we don't want to reabsorb here in the glomerulus because we're trying to take stuff out of the blood so that we can cleanse it. So we do not reabsorb at one end of the glomerulus. Let's go back and look at how your book presents this. We'll come back to that drawing, so keep it handy. Here is how your book shows it. Here's the afferent arteriole coming in. Here's the hydrostatic pressure of Bowman's capsule. Here's the osmotic pressure. I'm sorry, no, this is the hydrostatic pressure of the glomerulus. The osmotic pressure of the glomerulus and the hydrostatic pressure of Bowman's capsule. We add those all up and we get 10 mmHg as our net filtration pressure. Now, that tells us at any moment what is the pressure that the fluid is being filtered by. But what if we wanted to see how much liquid was being produced? How much filtrate were we producing in a given amount of time? Well, that is called the glomerular filtration rate, or the GFR. And we can find the glomerular filtration rate using this equation. It is the net filtration pressure times something called the filtration coefficient. And you do not need to know any of this. I'm not going to ask you to do these calculations. I just want you to know, what does GFR actually represent? This is how much liquid is being produced per minute. And it averages out to be about 180 liters per day of filtrate that is being produced. And that's about 60 times the volume of the plasma in your body that is being filtered every single day. Now, your book, the way it words it is a little bit weird. It says 60 times the volume of your blood, but it's actually 60 times the volume of your plasma that's being filtered every day. And GFR, using this equation, is directly proportional to NFP. Let's think about what that means. If NFP goes up, if there's greater pressure, then we produce more filtrate, so GFR goes up. If NFP goes down, then we produce less filtrate, so GFR goes down. When NFP goes up, GFR also goes up, and vice versa. When NFP goes down, GFR goes down. So that hydrostatic pressure in the glomerulus is going to be really, really important. We need it to remain relatively constant. We don't want it to fluctuate from that 10 mmHg very far at all. So the hydrostatic pressure in the glomerulus is actually independent of your systemic hydrostatic pressure or your blood pressure. We don't want, when your blood pressure goes up or down, for that to affect the hydrostatic pressure inside Bowman's capsule, inside the glomerulus, inside Bowman's capsule. Why? Because if NFP were to go too high, you can damage your kidneys. What you would end up seeing is those fenestration slits would actually get torn, and we would damage the glomerular membrane. But if NFP gets too low, then we're not going to filter our blood, and we're not going to remove all those potential toxins from your blood, all those metabolic wastes from your blood. And as that builds up, well, that could actually lead to death pretty quickly. So we need the hydrostatic pressure in the glomerulus to remain relatively constant, independent of the rest of your body's blood pressure. So how do we make sure that that NFP remains relatively constant? There's a few different ways. First, the kidneys can actually control that hydrostatic pressure on their own. It's something built into the kidneys. This is called intrinsic controls, or autoregulation. How do the kidneys control their own net filtration pressure? There's two different ways that we have of autoregulation. The first is the myogenic mechanism. Myo means muscle. Gen means created by. So the myogenic mechanism is created by the muscle built into those afferent arterioles. Remember, arterioles have a layer of smooth muscle. And let's think back to Bio 137 and something that we also mentioned back in the heart. Smooth muscle has a reflexive tendency to, when it is stretched, what does it do? It contracts. When you stretch muscle, any muscle, it will reflexively contract. So let's go back to our drawing that we looked at just a moment ago and see how does the myogenic mechanism work. Let's think about what happens if your blood pressure in your body goes up. Well, if we didn't do anything about it, the hydrostatic pressure of the glomerulus would also go up, and we could risk damaging the membrane. We don't want that. So if the hydrostatic pressure, if your blood pressure in your body goes up, as blood is coming into the afferent arteriole, is it going to be pressing harder or softer against the walls of the afferent arteriole? If your blood pressure is up, is it going to press harder or softer on the walls of the afferent arteriole? It would press harder. So let's see, how does our body account for that? As it presses harder against the walls of the afferent arteriole, that smooth muscle is going to reflexively contract, and we get vasoconstriction. So less blood gets into the glomerulus. Well, that blood is under higher pressure, but less blood is getting in, so that 60 mmHg hydrostatic pressure remains relatively constant, and the NFP can remain relatively constant. But let's look at what happens if the hydrostatic pressure of your body, your blood pressure, drops. Well, if the hydrostatic pressure in the rest of your blood drops, are we going to press harder or softer on the walls of the afferent arteriole? In this case, we're going to press softer. So the afferent arteriole now relaxes, it actually dilates to let more blood into the glomerulus. More blood's coming in, but that blood was under less pressure, so again, the hydrostatic pressure of the glomerulus remains relatively constant, and the NFP remains relatively constant. Reflexive activity of the muscles in the afferent arteriole constrict or dilate to let more or less blood in to the glomerulus in order to keep the hydrostatic pressure of the glomerulus about 60, and that keeps the net filtration pressure about 10. Okay, so here's that image that we keep seeing, and I've kind of alluded to the fact that we draw it this way because it's easy to point to certain areas and talk about a certain area, but that it's really important to keep in mind that it's not shaped this way at all. We're going to see now why it's important to keep that in mind. Right here at the top of the ascending limb of the loop of Henle, right where it meets the distal convoluted tubule, this area right here, in reality, sits right here in between the afferent and the efferent arteriole. They actually bump up against each other physically. And it looks more like this. We said this is what a nephron actually would look like. And here is where that's occurring. Here's the ascending limb, that elbow where it meets the distal convoluted tubule, and we can see that elbow is bumped up against the afferent and efferent arteriole. And again, we can see that right over here. So that's going to be important for our next type of autoregulation called the tubuloglomerular feedback mechanism. And that area that we just looked at, now we're looking at it in a little bit more detail. This is called the juxtaglomerular apparatus. The juxtaglomerular apparatus. And there's a few different cell types that are going to be really important for this tubuloglomerular feedback mechanism. First, here's that ascending limb of the loop of Henle bending right as it meets the DCT. We've got some cells right here along the wall of that tubule, and these cells are chemoreceptors. We give them the name the macula densa. The macula densa are modified chemoreceptors. Also, here's the afferent arteriole, and it's wrapped with some special type of cells called granular cells. Now, granular cells are all over the body. So we're going to be a little bit more specific. We're going to call these juxtaglomerular cells, or abbreviated JG cells. JG cells wrap around the afferent arteriole. Now these cells do a few things. One, they are stretch receptors. Two, these are the cells that release renin. Earlier we kept saying that the kidneys release renin. Now from this point on we're going to be more specific. The JG cells release renin. And the third type of cell of the juxtaglomerular apparatus are called mesangial cells. We won't really focus on those in this chapter. We will talk about the macula densa and the granular cells that we will call the JG cells. So we'll come back to that. But let's look at how the tubuloglomerular feedback mechanism works. First, I want to explain, we're going to talk about some stuff occurring that we haven't really discussed yet. We won't discuss it until later in this chapter. But if you come back and watch this part of this lecture at the end of the chapter, the things we're going to talk about right now will make a lot more sense. I want to go ahead and talk about it now, though, because it does pertain to filtration. So those chemoreceptors, the macula densa cells, what they are sensitive to is sodium and chloride concentration. These chemoreceptors detect the concentration of sodium and chloride. And based on the concentration of sodium and chloride, they will then adjust the diameter of the afferent arteriole. If you adjust the diameter of the afferent arteriole, what's that going to do? Well, if we let more blood into the glomerulus, net filtration pressure will go up and GFR will go up. If we let less blood into the glomerulus, NFP will go down and GFR will go down. So let's go back to our nephron here. If we let more blood into the glomerulus and we filter more, if GFR goes up, we make more filtrate. More filtrate then pushes through, so we're going to actually travel through the rest of the nephron faster. If we travel through the nephron faster, well, all along the way as we pass through, we're reabsorbing sodium and we're reabsorbing chloride. But if we're passing faster, we won't have as much time, so we won't reabsorb as much sodium and chloride. And when we get to this point right here, then there will be more sodium and chloride in the filtrate. Think about that. We went through faster. There wasn't as much time to reabsorb sodium and chloride. So when we get to right here, there will be more sodium and chloride left in the filtrate. The macula densa detects that. What will the macula densa do? It needs to slow the flow of filtrate. How can we slow the flow? Well, we constrict the afferent arteriole. Net filtration pressure drops. Glomerular filtration rate drops. We make less filtrate, so there's less pressure pushing through. The filtrate goes slower, and that gives us more time to reabsorb sodium and chloride. Once we get to this point, then we detect that the sodium and chloride levels have risen back. Have dropped back down. You get the idea. The rate of flow through the nephron is going to be directly related to the concentration of sodium and chloride. If it moves too fast, then sodium and chloride concentration will be high. If it moves too slow, then the sodium and chloride concentration will be low. And the macula densa cells detect that and adjust the diameter of the afferent arteriole accordingly. Here are those cells that detect that, and then they adjust the diameter of the afferent arteriole. So here that is in kind of graphic form. This is on page 899 of your textbook. If GFR is high, then there will be increased sodium and chloride concentration detected by the macula densa. The macula densa tells the afferent arteriole to constrict. Afferent arteriole constricts, we get reduced GFR. And then if this was low GFR, then there would be low sodium and chloride concentration detected. We would get dilation of the afferent arteriole, and GFR would be raised. So far, those two things were autoregulation. That was how does the kidney control its own net filtration pressure. But sometimes that pressure can be controlled by outside sources, something from outside the kidney, such as the sympathetic nervous system. If this arises from outside the kidney, we call this extrinsic controls. And the sympathetic nervous system takes over in certain cases such as hypovolemic shock. Let's use this as an example. If someone is hemorrhaging and they're losing a lot of blood, that activates their SNS. And your sympathetic nervous system responds, among other things, by constricting your afferent arteriole. It actually really constricts the afferent arterioles so that practically no blood gets into the glomerulus. This dramatically decreases the NFP and the GFR. But why would we want that? Let's think. If you are losing blood because you're hemorrhaging, do you really want to reduce your blood volume even further by filtering it? No. So temporarily your kidneys stop making urine. That's when sympathetic nervous system is activated. Now the other extrinsic control is the renin-angiotensin mechanism. Think about this. We've already talked about it back in the cardiovascular system. We're going to build on it now. Take a moment, and if you could, find that drawing that we did back in cardiovascular. We're going to take that same exact drawing, but we're going to add a little bit of detail to it. Here's some things that we're going to add. We're going to add a few things. Renin now is not just released by the kidneys. We have to be more specific. Renin is released by those JG cells, the juxtaglomerular cells. And originally we just said drop in blood pressure, but we're going to be more specific now. There are 3 things activated when there is a drop in blood pressure. Sympathetic nervous system stimulates the JG cells. Stimulation from the macula densa stimulates the JG cells. And a decrease in stretch. Well, if it was a drop in blood pressure, we've already said that we're stretching those afferent arterioles less. Those afferent arterioles are wrapped by JG cells, so we're stretching the JG cells less. When blood pressure is up, the JG cells are stretched and they don't do anything. When blood pressure is down, those JG cells are unstretched. They go slack and they start to release renin. So when there is a drop in blood pressure, all 3 of these things stimulate the JG cells to release renin. Now let's go back and pull out that drawing that we did earlier in the semester. Our renin-angiotensin mechanism, but we're going to expand on it. So let's just refresh. We said when there was a drop in blood pressure, when we talked about this the first time, the kidneys released renin. Renin took angiotensinogen and converted it to angiotensin 1. Angiotensin 1, now in circulation, it gets to the lungs where angiotensin converting enzyme, ACE, converts it to angiotensin 2. Angiotensin 2 had a fast, short-term way to raise your blood pressure by vasoconstriction, which increased peripheral resistance, increasing blood pressure. But it also had a slower, long-term way of raising blood pressure. Angiotensin 2 caused aldosterone to be released from the adrenal cortex, which caused sodium reabsorption, which leads to water reabsorption, increasing blood volume, and then ultimately raising blood pressure. Now we're going to add some details to this. First, renin, well, I'm sorry, I don't know what happened there. Renin from JG cells, not just from the kidneys. The drop in blood pressure is actually from SNS activation, from the macula densa, and from decreased stretch of JG cells. So when there's a drop in blood pressure, sympathetic nervous system, macula densa, and decreased stretch on JG cells stimulates renin to be released from the JG cells. Now let's go and add a little bit more detail over here. Aldosterone from the adrenal cortex actually causes sodium reabsorption and potassium secretion. How can one thing, aldosterone, really efficiently cause both sodium to move one way, reabsorption, and at the same time potassium to move the other way, secretion? What's something that moves sodium and potassium in opposite directions? Remember in Bio 137, the sodium-potassium pump? Well, the way aldosterone works is it tells the cells in your DCT and collecting duct to make sodium-potassium pumps. So we get sodium moving one way, potassium moving the other. But 3 sodiums are reabsorbed, 2 potassiums are secreted, which means more solutes are being reabsorbed. Now, more solutes being reabsorbed, solutes, what do? Solutes suck. They're going to pull water towards themselves. But that doesn't directly allow for water to be reabsorbed. All it is sodium is being reabsorbed. So let's look at something else that happens when there is a drop in blood pressure. Your hypothalamus, a structure in your brain, releases ADH. So there is ADH being released when there's a drop in blood pressure. ADH causes your kidney tubules in your DCT and your collecting duct to make these structures called aquaporins. Aquaporins are just little tubes that let water pass through. Specifically, ADH is going to make something called aquaporin type 2. We'll see what that means later on. But the aquaporins allow for water reabsorption. The aldosterone causes sodium reabsorption. That provides the pull for water. The ADH creates aquaporins. That creates the path for water. Both of those two things must happen in order for water reabsorption to be increased, leading to an increase in blood volume and ultimately an increase in blood pressure. Okay, so that is kind of summed up in this chart right here. Everything that goes on when there is a drop in systemic blood pressure. Lots of things happen, but they're all to maintain filtration. And that's where we will stop. Everything we've talked about in the first 2 lectures has really been occurring right here at the glomerulus inside Bowman's capsule. We've been filtering and seeing exactly how that takes place. Now that filtrate is going to enter the rest of the nephron, and as it passes through, we're going to process that filtrate. We're going to remove some stuff that our body wants to keep and place it back into our bloodstream, reabsorption. We're going to take some stuff from our blood that we didn't get rid of already and place it into the filtrate so that we can urinate it out, secretion. And that will be the focus of our next lecture or 2. Okay? Hopefully we've got the audio now and everything works. But otherwise, please let me know if you have any questions. If there's anything that I've not given to you guys that you think would be helpful, let me know. All right? Take care, stay safe, stay inside if that's a possibility for you, and I will talk to you next time.